Hey everyone, my name is Nancy Merino. I'm currently a postdoc at Livermore National Lab, and thanks for reviewing my presentation. Here I will be discussing how subsurface planktonic microbial communities can be used as a tool to track groundwater hydraulic connectivity and water quality. To do this work, we have a very multidisciplinary team made up of geochemists, hydrologists, and microbiologists from PNNL, USGS, Northwest Missouri State, and Desert Research Institute. To begin with, I want to discuss 3 reasons why microbes can be used as a tool. So those are concentration of microbes, the movement of microbes, and now we have cost-efficient methods available to do this work. So first off, microbes are present in significant concentrations in the subsurface, estimated at between 100 to 100 million cells per mil. The global average is 100,000 cells per mil. So these concentrations alone provide or suggest that microbes can provide valuable information on the inner workings of subsurface hydrobiogeochemistry. Second, microbes have been shown to move through subsurface materials, and several factors have been identified from previous studies, including adhesion processes that actually impede microbes' movement and may lead to the development of biofilms. There are also filtration processes, which essentially exclude microbes based on size. The physiological state of a microbe is also important and can influence the microbial cell size. So previous work showed that starved cells are actually smaller and can be more mobile through subsurface materials. The mobility of cells is also affected by whether or not the cell has flagella, and certain polymers on the cell surface can enhance or impede a cell's mobility. Abiotic factors also impact the movement of microbes in the subsurface. These include water flow rates and other hydrogeologic factors such as porosity of the material, hydraulic connectivity, and much more. Third, we have the tools and procedures to cost efficiently identify these microbes now. The procedure follows 4 overarching steps, from collecting the microbes to sequencing and data analysis. There are really nice programs out there in R, including ones that use machine learning to analyze microbial community sequencing data. And KBase actually now has the capability to analyze amplicon sequences in a super straightforward approach. And special shout out to Pamela Weisenhorn from Argonne National Lab who made this possible. There are many ways to analyze microbial community data. Once you get the amplicon sequences back, you can take a raw look at the data with relative abundance bar plots. You can do more comparative approaches with principal component analysis and network analysis. There are much more tools available out there, and in particular for subsurface microbial communities, we can also examine ecological null models to determine what ecological processes are influencing microbial community assembly. So these ecological null models help to determine whether microbial communities from different sites are more or less similar than expected by random chance alone. So for example, if we have 6 different microbes color-coded as 6 different colors, and we have 2 sites, site A and site B, biologists, so in one set of ecological processes, biologists call these deterministic or selection processes. And so in homogeneous selection, or for example, when you have similar geochemical conditions, we would expect the microbial communities to be similar, and that's because those microbes prefer to persist under those specific geochemical conditions. But under heterogeneous selection, where you have, for example, different geochemical conditions, then different microbial communities would arise. Next, we have stochastic processes, which are random impacts on the microbial communities. In the first set of stochastic processes, these are defined as dispersal processes. So in homogenizing dispersal, we would have, for example, fast enough flow rates or large enough pore sizes, and just ideally there would be very few hydrogeologic barriers. And so in this kind of ideal case scenario, microbes from site A can be dispersed to site B and potentially vice versa, depending on water flow direction. And in dispersal limitation, we would actually have the presence of hydrogeologic barriers, and so microbes cannot travel from site A to site B. In the second set of stochastic or random processes, we would have biological effects take place on the microbial communities. So this includes diversification with time, for example, with genetic mutations, or we would just have random drift, such as with birth, death, and reproduction. And so overall, these deterministic and stochastic processes can be determined by using the microbial community amplicon sequencing data. And these ecological null models, along with the other data analysis methods that I mentioned earlier, can provide a comprehensive analysis of the microbial communities. Okay, and so that ends the intro, and I want to just state this quote here, which was published in a paper in 2020, and it states that the microbial community profile in indigenous reservoir fluids is potentially a highly informative but still largely ignored source of data for reservoir engineers. And so this brings us to our case study where we wanted to examine the microbial communities found in the Death Valley Regional Flow System, or DVRFS. The DVRFS spans 100,000 squared kilometers, mostly in Nevada and a little bit in Southern California. It also includes the Nevada National Security Site, or NNSS, where hundreds of underground nuclear tests were performed. It also includes the Death Valley and Spring Mountains and just a wide range of altitudes, and it is a well-studied system, especially to understand radionuclide transport and groundwater levels. And so specific flow paths have been identified using hydrogeochemical data. And here I'm showing a map that is the latest hydrological model, and this was determined by one of the co-authors listed on this presentation, Tracy Jackson from the USGS. And so our specific question was, at this such large scale, can microbes inform us about hydraulic connectivity and water quality? And so we used DVRFS as our case study. Between 2008 and 2014, samples were collected from 36 different sites following the procedure that I mentioned earlier, and the data analysis that I'm actually describing here in this presentation has been submitted for peer review and is under the peer review process. However, if you want an earlier look at the data, you can look up this final report that I have imaged here. So I first took a look at the raw data with relative abundance bar plots. The x-axis has all the sites that we sampled at, and the y-axis is the relative abundance of each taxa in percent at the phylotaxonomic level. You can see some differences, but it's actually not really clear what's going on. So next I did non-metric multi-dimensional scaling in MDS, which actually suggests that microbial community varies by location. And NMDS essentially plots the microbial community sequencing data onto a 2D space. And here I'm showing two different approaches. One is the unifrac, which takes into account the presence-absence of a microbe, and Part B, weighted unifrac, takes into account the relative abundances. And each point in these plots are different sites that were sampled at. So let's plot this alongside the map. And first we can see that Spring Mountains and Ash Meadows have similar microbial communities. Next we can see that in the upper groundwater basin of DVRFS, the Paiute Mesa and Oasis Valley communities are actually different, and Oasis Valley communities actually seem to be pretty unique. So stay tuned for more on this discussion, or you can just skip ahead to the end. NNS sites and sites nearby NNS seem to have pretty similar microbial communities. However, there should be some hydrogeologic boundaries, or there could be interbasin groundwater flow. So we have to dig in deeper to understand what's going on here. And I did some ecological null models and network analyses, which we will discuss later in this presentation, which actually suggests that there are some differences between these microbial communities. Finally, we can see that Death Valley is somewhat similar to sites within and nearby the NNSS. And this is actually super important because there are two different models for groundwater flow in this area. The first suggests that that water flows from Ash Meadows to Death Valley through deep carbonate rock, known as the deep carbonate flow conceptualization. However, the current model suggests that water flows from Amargosa Valley to Death Valley through shallow basin fill, known as the basin fill flow conceptualization. So now the question is, does the microbiome actually provide more evidence for basin fill flow conceptualization? More data analysis is needed, so stay tuned. And so overall, we can see that from NMDS, we have 4 overall groupings of the microbial communities. So next I did network analysis of the common microbes, which suggests also that the microbial communities vary by location. Here the common microbes are defined as those with low coefficient of variation across samples and were observed at least 2 times in more than 2 samples. So each circle here represents each individual microbe that was identified. The size of the circle represents the number of counts, and each cluster is labeled and identified by color. The donut charts around each cluster represent the ratio of microbes per location for each cluster. And overall, we can see that common microbes are clustering by location. So from the network, we can see that common microbes from spring mountains and ash meadows are connected in 2 clusters. And importantly, ash meadows clusters is actually only connected to the network via the spring mountains cluster. These 2 sites are also separated into 2 clusters, which indicates there is some degree of separation of the microbial communities between these 2 sites, and this was further corroborated by ecological null model analyses. And also stay tuned for more discussion on unique microbiome of Oasis Valley. Next, we can see that Amargosa Valley is actually kind of the center of the network and is a mix of microbial types, likely representing the mix of groundwater sources that transect this section. I've listed some names at the bottom in the gray box for anyone interested to enjoy what microbes are found in Amargosa Valley, French Mayuka Flat, Rainier Mesa, and so, feel free to pause the video and just read the names because there's some super interesting bugs. There may also be some connection from Paiute Mesa to Amargosa Valley, but we definitely need more samples to corroborate this assessment. And finally, we can see that the Death Valley cluster of common microbes is closely associated to the Amargosa Valley cluster of common microbes, and along with ecological null model analyses and a previous report on the microbiology of DVRFS, this suggests that the microbiome does support the basin fill flow conceptualization. So finally, let's get to the elephant in the room and discuss why the Oasis Valley microbial communities are so different than the other microbial communities of the DVRFS. I have to caveat this and say that we had limited samples for Oasis Valley. We only had 2, but from this data, we were able to develop 2 hypotheses. So the first hypothesis of why Oasis Valley microbial communities are different is that Oasis Valley is kind of isolated and that there are biological barriers, or there actually may be some contamination from surface water microbes. And so there may be also slow water flow from Paiute Mesa to Oasis Valley, so the microbial communities are not really reflecting each other. And there might also be clogged pore spaces or just different geological confining units with different hydrologic properties preventing the dispersal of microbes through this groundwater basin. There may also be infiltration of surface microbes into the groundwater system, as we observed many aerobic and sediment and soil-related microbes in Oasis Valley. The second hypothesis is that Oasis Valley communities represent kind of pristine community in this groundwater basin, where Paiute Mesa communities have been impacted by underground nuclear tests and likely represent a reassembled community adapted to its new environment. There could be effects from radiation, mostly when the underground nuclear tests were performed. There could also be an increase in hydrogen and carbon dioxide from radiolytic and seismic activity. And seismic activity and earthquakes have been shown to actually perturb microbial communities. And so Oasis Valley could represent kind of an unperturbed community in this location. And so this groundwater basin is actually super ideal for future studies. And we could have artifacts from our study. And so we need to actually collect more samples along this flow path to further understand these 2 hypotheses and understand the microbiome of this groundwater basin from Paiute Mesa to Oasis Valley. And so in conclusion, from this case study of DVRFS, and in my opinion, we need to have hydrobiogeochemical data in order to comprehensively understand groundwater systems. We have more advanced tools out there now to study the microbial functions, the activity, and the metabolites. Here I'm just showing amplicon sequencing data. And so, all of this combined, when we combine all the microbial community tools out there available, and we can combine it into reactive transport models or simulations, we can really deeply understand subsurface environments and groundwater systems. Finally, I'd like to acknowledge all the people and funding sources that went into making this case study possible. This is copied and pasted from our submitted manuscript, and I'm still early career, but this is quite possibly the longest acknowledgments I've seen out there. There's just a lot of people and funding that went into making this possible. So thank you so much for listening. Here's my contact info and data availability. Hopefully our peer review process goes smoothly. And I'm looking forward to any questions that you guys have, and see you at the conference. Bye.